Hi, my name is Ishita,
Hi, my name is Rajika.
and we're presenting on mapping eye color mutations in Drosophila. So we were given two unknown mutations called LOT and TRI, both of which were recessive. The goal of this experiment, there were two main goals of this experiment. One was to identify which mutation was X-linked and which mutation was autosomal in terms of their mode of inheritance. And the second goal was to determine where that mutation was in terms of what chromosome number it was on, as well as which gene that mutation was on. So here is an introduction to those mutations in a visual sense. So you can see that the lot mutant was characterized to have a brighter orange eyes in the flies, and the wild type had darker, a little redder eyes with black spots. And finally, the tri-mutant was characterized by having a black eye color. So as you can see, all the phenotypes are shown in the color of the eye.
So the first experiment we conducted was a cross to determine if either of the mutations was X-linked or autosomal. So both of these experiments were conducted the same type of cross and involved crossing a female mutant with a wild type male. The reason we did it this way was because we could control so that the results in the F1 generation were indicative of whether the mutant at hand was X-linked or autosomal. So if the mutant was X-linked, we expected to see that in the F1 generation, all males would be expressing the mutant phenotype and all females would be wild type. We expected this because for X-linked traits, males would only inherit the allele from the mutant mother, and females would inherit a wild type allele from the wild type father and express that trait. And then if the allele was autosomal, we expected that all of the F1 offspring would inherit two copies of the gene and would inherit one wild type from the father and one mutant allele from the mother, resulting in all of the F1 generation flies being wild type. So in order to set up the cross, we used virgin mutant females. And it was important that we use virgins because Drosophila females carry sperm from any previous mating with them throughout their entire lives. So if these females had already crossed with other mutant males, this would mess up the results of our cross being conducted with the wild type males, and we wouldn't be able to clearly predict whether the allele was the allele was X-linked or autosomal. So we collected the virgin females by clearing out all adult flies from a 9 to 11 day culture, and we waited for females to eclose out of this vial. We knew that these females would have to be virgin because males are not sexually mature within 6 hours of eclosure. And we then mixed together 7 to 10 virgin mutant females and 7 to 10 wild type males in a new vial to conduct our P1 cross.
So here are results from that said class that Rajika was talking about. So as you can see, for the tri mutation, we had both wild type mostly for males and females, which, as she said, suggests an autosomal mutation or autosomal mode of inheritance for the tri mutation. However, you can see that there were two tri mutated males and three tri mutated females, and this might be because we waited between 6 and 7 hours instead of less than 6 hours before we examined or before we took out the female virgin flies. And what this would result in is perhaps there was a male fly that became sexually mature and impregnated one of the virgin or so-called virgin female flies, thus sort of messing with the results a little bit. In the second table, you can see that the lot mutation has an X-linked mode of inheritance, and this is because all of the males were mutated and all of the females were wild type.
So the next experiment we conducted was 3-point mapping with the triallele, and we received simulated data to determine where the tri mutation was located relative to two other genes on the chromosome 3, which we were told the tri mutant is located on. So in order to conduct a 3-point mapping cross, we crossed a heterozygous female with a homozygous recessive male. This means that the female was a heterozygote for all three of the genes under study, and the male was homozygous recessive for all three of the genes under study.
So as you can see, this is our data from that F2 generation. It is important to note that this was simulated data since we were not able to do this physically in the lab. So as you can see, the number of flies where they were the greatest, this suggests non-crossover in these flies here, But in the bottom two rows, you can see since they have the smallest number of flies with these phenotypes, that suggests that these represent double crossovers in the actual gene sequence. So using this as well as the single crossovers in terms of the phenotypes, we were able to determine a general sequence of these mutations. And so using those mutations, we could then begin to attempt to map the tri mutation specifically.
So this was a genetic map we came up with based on our data. So like Ashita said, we looked at the recombination frequency in the F2 generation to figure out how far apart certain genes were from each other. So for SB and Li, we found that 16.57% of all of that F2 generation offspring had combinations of those two genes that weren't present in the original parents. So we put them at that distance apart from each other. And then for Li and Tri, we also found that about 13% of the offspring in the F2 generation had novel combinations. And so we made the predictions for the distance based on that recombination frequency. We also knew that Li was in between SB and Tri because, like Ashita said, it was the one gene that remained in place when both SB and Tri were switched in the no crossover and double crossover groups. So our next step was to make a hypothesis about the identity of the unknown. So where was the tri mutation actually located? So we know that the tri mutation is on chromosome 3, and we were also given the fact that LY and SB were located at specific locations on this chromosome. So now knowing the distance of tri from LY and SB using our genetic map, we were able to estimate that tri would be about at region 27.45 map units from 0 on chromosome 3. This would place it closest to gene SE, which is located at 26.0 map units from 0. And gene SE codes for sepia eye color. So this corresponds to the phenotype that we observed in our tri-mutants, which was a darker sepia eye tone.
And this, now we want to talk about what our results might have looked like if we had performed a complementation analysis with the flies. So we would have chosen to cross recessive tri flies with recessive mutant flies. And these other mutations would be on several suspected genes, which we know which genes those mutations are on. And how our results might have looked like were if the tri mutation and the mutation on a certain gene, if that gene happened to be where the tri mutation was also on, then we would have had mutant offspring. Whereas if the mutation and then the tri mutation were on different genes, then they would be having wild type offspring. So based off of which offsprings were mutant and which offsprings were wild type, we could determine which gene tri was on.
So essentially, some of our results, we conducted a cross between recessive mutant females and wild type males to determine whether lot and tri were X-linked and autosomal. We found that tri was autosomal and that the lot mutation was X-linked. We then conducted a 3-point cross using simulated data to determine where tri was located relative to two other genes on chromosome 3. And then knowing the location of those two genes on chromosome 3, we were able to predict where the tri mutation was located on chromosome 3, and we put it closest to the SE gene. And we would have conducted a complementation test to determine whether tri is in fact located in the SE gene.
So in terms of where we can go with this experiment, Rajika and I were looking more into the sepia gene, and we saw that it codes for the protein pyrimidodiazepine synthase. So we were wondering if perhaps the tri mutation resulted in a null phenotype where this protein is not functioning. So potentially we could perform a Western blot under flies or under tri mutants and under wild type flies to see if the protein was present in those tri mutants using the Western blot data. Thank you.
Thank you.
That concludes our presentation.